I think it's recording. I never know, so I kind of delay this beginning part. Okay, let's start. So, this is going to be my real run through, as if I was pretending I was at OAS. So I have no notes. Well, I do have one little note card that I think I'll need, but we'll see. And I'm starting the timer. Hopefully this is under 15 minutes. Crossing my fingers. Okay. Hello everybody, my name is Ruby Perez, and I want to thank you all for joining me to figure out if dopamine signaling changes in a Huntington's brain. So some background on Huntington's disease before we dive into the study here is it is a neurodegenerative disease, and this disease is fatal. So let's look at a brain here. This is a human brain, and it is healthy. There's nothing to point out here. It's a good-looking brain. But this is a brain with Huntington's disease, and as you can see, the difference between the two, the size is much smaller. The holes in the middle, called ventricles, where your cerebral spinal fluid flows, is much larger, and you have these gaps. You can see them most noticeably at the top, right down the middle. The gaps are bigger. You just have an overall loss of brain tissue. So we'll get into what's going on just a bit, but how does someone get Huntington's disease? Well, Huntington's disease is purely genetic, and it follows an autosomal dominant inheritance pattern. And this figure here shows that the father of this family has the condition, so the father is affected with Huntington's disease, while the mother is not. And they have 4 children. The likelihood that their children will inherit the disease is 50%. So every time they have a child, there is a 50% chance that the child will inherit the disease. And if the child has the gene that causes the disease, then they will show symptoms. So the gene in question is called the Huntington gene. Who would have thought? So off to the side, we see the gene and what it looks like, the location of the gene, and the top gene is normal. There are no mutations, and you can see the red are the cytosine, adenine, guanine nucleotide repeats that make up the gene. When you have too many of these repeats, these CAG repeats, you can see that it gets tangled up and it looks like just a mess. So when you have too many of these repeats, it becomes very problematic. And as you saw from the first photo, the brain is shrinking. This is because these inclusions, these tangles of the protein that make the gene, or the protein from the gene, become sticky, and they stick to the cells, and they're toxic to the cells, ultimately causing cellular suicide. And so these cells are dying, these neurons are dying in the brain. And so what do we see from this? Well, there are 3 main categories that happen in Huntington's disease. We start off with, no, we don't start off. One of the categories is cognitive symptoms, and these include things like a decrease in working memory, dementia, problems along those sorts. We then have another major category of motor symptoms, and these symptoms include not being able to control your limbs, your facial expressions. A lot of people have problems swallowing and chewing, and that is something that happens that causes them to die. They also have problems controlling their diaphragm, so these motor problems are very problematic. But what is less known is that Huntington's patients actually show psychiatric symptoms about 10 years before the motor symptoms show. And the motor symptoms are typically what send people into the clinic to be formally diagnosed with Huntington's disease. And as is with any disease, there are a wide range of psychiatric symptoms that happen, but the ones of the focus today will be impulsivity, risky decision-making, and the perseveration or the continuation of these 2 behaviors. So to look at impulsivity, risky decision-making, and perseveration, we're looking at a very specific brain circuit, and that is the mesolimbic cortical pathway. This circuit is of interest to us because of two specific nuclei that receive dopamine from the brainstem. We've got the nucleus accumbens and the prefrontal cortex. These brain regions both receive dopamine from the ventral tegmental area, and dopamine is very important in this circuit. However, dopamine is not the only neurotransmitter involved in this circuit, which adds to the layer of complexity in trying to answer the question if dopamine signaling changes. There are other modulatory neurotransmitters affecting dopamine. So, to do this, we decided to look in a mouse model, specifically the N17182Q mouse model. Mice are genetically similar to us, so we're able to make inferences about their biology and compare it to our biology. And this mouse model is special to our study because they show symptoms similar to those I'm going to practice a lot more, I promise. They show symptoms similar to adult onset Huntington's disease. Their progression is slower than other relative mouse models that have Huntington's disease. And also they're special because they have two of their own Huntington genes, but they also have a fragment of a human mutated Huntington gene. So that's adding to their symptomology. So from this, my hypothesis becomes, back up here. So, dang it. Yeah, there's a problem here. So my hypothesis becomes those with Huntington's disease will have less tyrosine hydroxylase or dopamine intensity in the mesolimbic cortical circuit as the disease progresses when compared to healthy controls. Just kidding, there was no mess up. I'm overreacting here. Okay. So I had just said tyrosine hydroxylase, and you might be like, what did I miss? You didn't miss anything. Tyrosine hydroxylase is the rate-limiting enzyme of dopamine, and here you see the dopamine cascade in the presynaptic neuron, and there circled in green is tyrosine hydroxylase. Tyrosine hydroxylase was chosen to look at over dopamine because tyrosine hydroxylase is more stable for a longer time than dopamine is, because dopamine has other things that are degrading it, whereas tyrosine hydroxylase is a little better protected. So the amount of tyrosine hydroxylase indirectly tells us how much dopamine is in the presynaptic neuron. So the methods can be looked at on a timeline. So I mentioned the mice. When we have a grouping here, there were two time periods of our mice. We have 5 weeks and 15 weeks. So all the mice were born at the same time, but about half of them were sacrificed at 5 weeks of their life. The other half were sacrificed at 15 weeks. And half of those groups were control mice, and the other half were Huntington's mice. So we have our 4 groups. After the mice were sacrificed, we treated their brain tissue using standard immunohistochemical techniques, immunofluorescent techniques that allowed us to take photomicrographs, like you see here on the right, and that allowed us to make computer analysis using a software that measures intensity. And what we were looking at is the more intense a region is, the more dopamine is there. This is a special case because what we see is, I don't even know if I should say that. Okay, so back to our mesolimbic cortical circuit. This region outlined in yellow here is the nucleus accumbens, part of the nucleus accumbens. So the nucleus accumbens can actually be broken down into 2 subregions, the core and the shell. The outline here is specifically the core, and the core has been kind of coined by Stan Floresco as the go-to nucleus, and this nucleus is involved in recruiting other motor regions to physically go toward a stimulus. I will mention the striatum as well, because we looked at the striatum while we were looking at the core. The striatum is different, however, because the striatum is part of the basal ganglia and a different circuit, and essentially it does the same thing, so we thought we would see similar results. We did see similar results, but they were not consistent with our hypothesis. So dopamine signaling did not decrease in either the core or the shell. So we can see here to the graph on the left, the red line represents the mutant category and the blue line represents the wild type category. And while it looks like something interesting is happening, those at 5 weeks are not any different than those at 15 weeks, and mutants are not different than the wild types. And again, the same happens in the striatum. The blue line represents the control wild-type mice, the red represents the mutant mice, and while it looks like something might be happening, there is no difference between the two. Moving on, I had mentioned that one of the regions we were looking at was the prefrontal cortex. The anterior cingulate cortex is part of the prefrontal cortex, and it is involved in decision-making, emotional regulation, and error detection. So, let's see if dopamine signaling decreased. It did. We had a main effect for time, which is not exactly what we were hypothesizing, but it's part of what we were hypothesizing. So there's something about being a 15-week-old mouse that makes you different than being a 5-week-old mouse in terms of dopamine signaling. So 5 weeks had more dopamine than 15 weeks. We're losing dopamine as we age. Again, back to the nucleus accumbens. I said there were two parts. The second part is the shell, and the shell acts as the brake, where the core is kind of the gas pedal, the shell is the brake, and it is a suppressor of behaviors. So let's see if dopamine signaling decreased. It did. So right here we have the blue line on top, the wild types control. Those mice have significantly more dopamine than the mutant mice, suggesting that there might be something about being a mutant that causes you to have less dopamine in your brain, specifically in the shell of the nucleus accumbens. So again, to reiterate, the core and the striatum had no difference, and that was not hypothesized. The anterior cingulate cortex had a difference for time such that those at 5 weeks had more dopamine than those at 15 weeks, irregardless of what group they were in for their genotype, which might suggest that their dopamine loss is due to normal aging. Those in the shell, however, had less dopamine than the wild types in the nucleus accumbens. The mutants had less dopamine than the wild types. And this is consistent with our hypothesis. So, there might be 3 explanations as to why, and they go back to those personality changes that we were seeing, that we were looking for at the beginning. So, in regards to impulsivity, it has been noted that a decrease of shell activity, or shell signaling, dopamine signaling, has been correlated with an increase of inappropriate responding. So an example of this might be, let's say you're giving a presentation and they call your name up to the podium to give your presentation. An impulsive move would be to stand up to the podium before your name is even called, or to take the microphone away before the person is ready to give it off to you. An example or an explanation as to why riskiness might play, or a decrease of the shell, a decrease of dopamine signaling in the shell relates to riskiness, such that the shell plays a role in learning to inhibit behaviors associated with with punishment. So an example to this might be gambling. When you lose money, it's kind of a punishment. It's not kind of, it is a punishment. And if your shell is deactivated, then you can't learn to inhibit these behaviors that are causing this punishing consequence, losing money. And it is known that a lot of people with Huntington's disease have problems with gambling. They lose a lot of money They're impulsive to buy things as well. So that plays a role. On to perseveration. A decrease of dopamine in the shell has also been shown that a decrease of dopamine in the shell has been related to perseveration, whereas behaviors that are continuous, or where people perseverate, continue long after rewards have been removed or punishments have been applied. So these people make these impulsive and risky decisions, and they continue to make it. And the shell might be one of the suspects as to why. Some limitations. So the time that we looked at for each animal didn't give us a complete answer as to what was going on. I had noted that the anterior cingulate cortex was decreasing for time, but we don't know where the two genotypes converge from each other. So looking at a time period beyond the 15 weeks might help us see where exactly these two are changing, which might allow us to make claims about the rate of decline. Unfortunately, with this mouse model, they don't live very long. They, if you're lucky, might live into around 25 weeks, 28 weeks, but after that they start dying rapidly. It's sudden. Sort of a limitation, but also a future direction, would be to run behavioral tasks on these animals to be able to make behavioral claims. We were not able to do that during this study because we did not have enough time. We did not have the resources to, but that would be something that would supplement our results as well. A future direction for this research would be to look at the receptors. So this picture came up before when I was noting out tyrosine hydroxylase. I'm showing it again because the bottom half of the photo we see this postsynaptic neuron, and it has this receptor on it. And we didn't think about the receptor. Well, we thought about the receptors. We're thinking that the receptors are having a compensatory effect to the release of dopamine. So it might be that dopamine signaling is perfectly fine, but the receptors are degrading. Or you might see, dang it, I messed that up. So yeah, dopamine receptors might be changing, whereas the dopamine signaling itself is remaining the same. And that was not what we were able to see with our particular staining techniques. So in the future, that could be something that we do instead. However, the take-home message for all of this is to think of the circuit as a whole. Neuroscience research is very difficult because there are so many components that could play into why something is not working. You look at this simple electrical circuit here, you have a basic switch, battery, and light bulb. If one of the things is broken, you can easily point it out by examining the circuit. However, you might not know why the circuit is not working if every little component of the circuit is slightly degrading. Slightly degrading, maybe not significantly degrading. So there might be a summation of degradation that is happening that we are unable to see in the brain. And with that, I would like to thank you, audience, for listening to my talk. I would like to thank Zach Daniels and Dr. Mark Pitzer for many months in the lab working and looking at mice brains. I would like to thank the McBride Lab at the Oregon National Primate Research Center associated with OHSU and the mice themselves, along with Dr. Zachary Simmons for assisting along the way. And here will be my question slide. Questions, comments, concerns. And these are if people are like, what about the environment? Why that mouse model? So, okay, that is it. Pretty shaky.